Good morning, everyone. It gives me a great pleasure today to introduce Dr. Bob Ferris, who is a world-renowned immune therapy expert and cancer immune therapy expert. You probably know him in that capacity, but you probably know Bob as well because he's been here multiple times doing grand rounds, at least probably 3 times, I would guess, in addition to being a guest at our several head and neck cancer symposiums that we've had over the years. Bob had trained initially at Hopkins. He received his medical degree and PhD in immunology and did his residency in otolaryngology and head and neck surgery, and subsequently trained in head and neck oncologic surgery. And then he moved to University of Pittsburgh around 2002, where he quickly established his immunology lab, and in 2007 became the co-leader of the cancer immunology program at the University of Pittsburgh. Around the same time as well, he became the vice chair of clinical operations in the Department of Otolaryngology. He also, interestingly, has other titles, including being a professor in radiation oncology. And yesterday we were touching a little bit on some aspects of radiation techniques, but Bob promised that that will be the topic of his next grand rounds here at Winship. At the international and national level, he has also been very prominent over the last years. He has been the program chair at the Quadrennial International Conference for the HNS meeting in Seattle back in July, and will be also the chair for the future conference. At UPMC, he has also been Associate Director for Translational Research since 2012. He has been a member and chair of multiple national committees on head and neck cancer. He's a member of the Head and Neck Cancers NCI Steering Committee, has chaired the previous PULA task force, and has served also as co-chair of the Econ Head and Neck Cancer Committee. Suffice it to say that the work of Bob and others in the field has really paved a new era in cancer treatment. A lot of us basically are currently immune therapy or use immune therapy, and some of us basically claim to be immune therapy experts, but I think people like Bob have been doing this for such a long time, and really it's great to have someone who can combine the immune therapy world with the oncology world like Bob has done, especially with head and neck cancer. So without any future delay, it's my great pleasure to welcome him again in Atlanta. Thank you, Bob.
This live? So it's the first IQ test of the day, is can I function, can I work the microphone? Nabil, thank you so much. What a kind introduction. And Nabil mentioned that I've been here before, and so it's been one of my goals to always have something new so that I get invited back, because I have such a great time, and it's a challenge to have enough new stuff. Many of you may know my Dad went to dental school here in the early 60s. My sister went to college here and lives up in Marietta, and my mom as well. My mom is bionic now. You guys gave her a new back, a new knee, and then last week a new hip. So whether I get frequent flyer points for grand rounds or she gets them for, keeping the lights on and keeping the ortho department going, I don't know, but she's always done really very well, so I know the medical care here is great. I tried to put together a talk that synthesized a little bit of what, for a while in my career, were 2 different groups, different meetings, the group of us in the immunology world and then those of us in head and neck cancer. And so hopefully by the end of it, you'll see how they have really kind of merged together, and tell you a bit of a story about how a surgeon can integrate oneself into a prospective oncology field to try to collaborate and build teams to try to help patients. So I have to give you my disclosures. Our departmental motto in Pittsburgh in otolaryngology is to heal with steel. So we do that, until we give immunosurgery. We also have a hockey team, you may know that, and I serve on the medical staff of the Penguins, so I have to disclose that. Got to touch the cup, and my kids were eating Skittles out of it last year, which kind of grossed me out, but anyway, we've got the cup for another couple months, so we're hopeful. So I think any good translational and clinical research starts with a patient problem, and this is the patient, kind of person who was coming in the 90s. Those of us seeing a lot of head and neck cancer patients noticed this patient who really had really no traditional risk factors, was not a big kind of smoker, drinker patient. Had a neck mass. We used to say this guy would notice it while shaving, but I think he kind of skipped a couple days here, but he noticed it one way or another. And this is a unique neck mass, kind of cystic, fluid-filled, and would often be kind of shucked out by the surgeons in the community thinking it was a congenital cyst, and then all of a sudden the pathologist told them they had a cancer patient on their hands. And so this person is younger. The typical head and neck cancer patient for years was 60, not 50. Very asymptomatic, no obvious primary tumor. We'll get to that a little bit. And the interesting thing is we discovered a new disease in our midst. And this HPV positive disease has been rising at about a rate of 300% over the past 20 or 30 years, so a major clinical problem. And as you may know, the AJCC staging system was just revised. The 8th edition came out January 1, 2017, and overnight, this patient went from stage 4 to stage 1. So this gives you a sense of a clinical problem that we need to step up as the scientists and the investigators to figure out what is the best way to stage and treat and then follow such a patient. And this is really a phenomenal, if you just stop and think about it, to have a new disease arise in the context of an oncologic career is pretty amazing and exhilarating. So fortunately, public health efforts have gotten smoking to get down to about the 20% level. This was 2005. My understanding is down to about 17% of all adults. So the smoking-related head and neck cancers have been declining along with this. But interestingly, the overall aggregate head and neck cancers are going up because of this HPV positive cancer that has been overtaking the decline in smoking-related cancers, so that the aggregate head and neck population increases, and of course the demographics change. We have hedge fund managers. We have a few years ago at our meeting, Michael Douglas came and spoke at the American Head and Neck Society, and so the public is becoming aware of this issue, and that changes the kind of patient visits that we have in the office. But what really kind of triggered me was, besides that you have a big clinical mass of patients, is that we really don't know how to treat them. And I would suggest that these are all standard of care treatments that a patient may get, and the decision will be made simply by where that patient lives and decides to walk into the clinic. That there is a convention in each institution, bias, if you will, which is not always data-driven. So I would say concurrent chemoradiation has been the standard of care for 20 years for cancers of the tonsil and the base of the tongue where HPV arises. Cetuximab plus radiation is an FDA approved since 2006, so you've got 2 different FDA approved regimens. The folks at MD Anderson and perhaps here use radiation alone for low volume disease. Us surgeons feel like we've been operating on head and neck cancers for decades, and if we operate up front, then we can decide post-operatively the extent of radiation or chemo. And then we have level 1 data with induction chemotherapy, the TAX-324 trial, followed by concurrent chemoradiation. And then, there's a part of you that says it's a virus. In fact, I had a patient 5 or 6 years ago that had an HPV positive head and neck cancer, turned out to be friends with the president of UPMC, the big UPMC, 62,000 employees, and I would get summoned down to the US Steel Tower to explain to him why I was not using an antiviral on his patient, on his friend. And shouldn't we be using immunotherapy for a virus-induced cancer? So I'd like to go through these, and I'll end with the immunotherapy, but I would just challenge you to think, how would this patient be treated here? Because do we have the data for these 5 options? Because patients ask us, what's best, doc? And we need to generate the data to answer that question. So the first important thing to know is that, as you've probably already heard, HPV positive patients do better than HPV negative head and neck cancer patients. And the seminal paper in this regard came from the Radiation Therapy Oncology group. I think you guys are all probably aware of the importance of this cooperative group in our field. And Mara Gillison and Kian Ang put together an HPV risk stratification based on this trial, the RTOG 0129. And I think all I would kind of clarify for you is that we can take a population unselected and using HPV, tumor bulk, and smoking status, segregate them into low risk, intermediate risk, and high risk. That's one of the key features to figure out whether we should be, what's the extent of treatment we should be giving and whether we should be giving combined modality therapy. So what you have here is a low risk group, never smokers, low volume disease that have survival of 90 to 95%. That's quite amazing. You have a group of 70% or so, 65 to 70% who have intermediate tumor bulk or are smokers, and then any HPV negative patient is still down in the 40 to 50% range, really hasn't improved in years. So using a conceptual concept like this, we can then put patients into prospective research to, and we've coined the term in the field, to de-intensify therapy for the low risk, or to intensify therapy, not just by adding chemotherapeutics, but being a bit more targeted and understanding the biology for the HPV negative tumors. And this was driven by some data that Mitch McTay published. He's at Case Western, and you can see he aggregated the late long-term toxicities of a few of these RTOG trials where the survival curves may have looked good, but we asked the question, what's the function and quality of life? In the long-term morbidity from that first option of concurrent chemoradiation really appeared to be suboptimal. Although we were curing some of these patients, many of these patients, they had severe problems long-term, and so the clinical problem is you have a younger patient and yet they have to live with the toxicities of therapy, so we need clinical research to drive the appropriate treatment choices. So the first trial to address this, targeting specifically this new disease of HPV positive patients, was the Econ 1308 trial. This was just published a month or two ago. And this trial asked, if you only enroll HPV positive patients, can you get away with lower dose radiation therapy? Can you de-intensify using a upfront selection process of induction chemotherapy? So these patients, only 90, it was a small trial of cisplatin, paclitaxel, and cetuximab. And if they had a complete response, which about 2/3, 65 to 70% did, they could get away with 54 gray instead of 70 gray of radiation. So the data for whether 15 gray is really functionally impactful, I think is still not out there, but we think that if you can maintain survival with less radiation, long-term effects should be better. And so this was actually the interesting result, that in those patients who had this intermediate bulk and were never smokers, you could get around 95, 96% disease-free and overall survival. And it's been said that this one fellow here had such good quality of life, he actually died while whitewater rafting, and that perhaps maybe a little bit less quality of life might have been useful in that regard. But we say that, of course, feeling bad that this happened, but he actually was doing quite well at the time. So then you have the RTOG, which was new in the newly aggregated NRG cooperative group. And this HN002 trial asked a different question. Not can you give induction chemotherapy, but can you use that low volume disease, never smoker group to enroll a population of patients to get radiation alone at lower dose, 60 gray instead of 70 gray, with or without weekly chemotherapy, instead of the high dose chemotherapy that may be more toxic. This trial accrued extremely rapidly. The RTOG Head and Neck Committee is quite good at enrolling to these trials, and of course, there's many HPV positive patients. So this is now completed, and everybody is anxious to see where it's going to go. Oh, please. No, no problem.
This is a good challenge, like in Merkel cell and other viral cancers, to take like what people think are curative paradigms and change that paradigm. And so you mentioned how you evolve really quickly, but I find sometimes it's really hard when we're designing trials to say, look, we're going to do something different than what people think are curative.
So yeah, so that's a question not only for the doctors who are concerned that de-intensification would take that 90-95% survival and maybe end up at the end reducing it. And it's a hard thing, as you, I think, are alluding to counsel patients, to put them onto a regimen, and what is the equipoise on the part of the treatment team to explain de-intensification to a group of patients, because you're sort of, and we do this every day. When I walk in and have the patient has like Dr. Google printed out, you just pull up a chair because it's going to be a half-hour appointment, but you're trying to tell them that they may have these toxicities, and so you're trying to avoid something they may have, but they don't currently have it. And often patients think, oh, it won't happen to me, and they often say, in some trials I'll show you, I've had people go off trial because they don't want to be de-intensified when the push comes to shove. Some of them, the spouse is a physician or in medicine, and they say, no, throw the kitchen sink at it, you got it. So it is a challenge, and I think we can still enroll very quickly, even though some patients are uncomfortable with it. The problem I've had is when they get on the trial and then they want to bail on step 2, and that's a challenge. But that's why we over accrue. But there are so many of these patients that we can enroll still quickly, even with those caveats. And I would suggest that even though we have data safety monitoring committees that would stop the trial for certain parameters if there were too many recurrences, and we build this into each trial, the high volume centers know, and with each successive accrual know the patient's doing well on this trial. And so if we were starting to see some signals, even without a national meeting or DSMC, the word gets around pretty quickly if it's unsafe.
And there were early stopping rules on some of these treatment
Right.
treatment reduction costs.
Exactly. So I set this up because you need to understand where the non-surgical regimens are going, even though we don't have the results of this trial, to understand where the surgeons were, to try to figure out how to contribute to this field. In part because for decades, the surgeon brought to the multidisciplinary treatment team accurate staging. This was sort of the key, the radiographic imaging for head and neck, whether the lymph nodes were positive, whether there is extranodal extension, one of the worst prognostic factors in the disease, was present. You had to sort of do the neck dissection. And, a few months ago, we hit the 10th anniversary of the iPhone. So technology has improved for surgery. We have better visualization, better access, better margin control. And so we have the ability through the laser and the robot to get a tumor out without a morbid operation. We can remove lymph nodes without having to remove important structures. And yet the surgeons had to evolve our practice to try to convert an open procedure where we all trained in the 80s and 90s to approach the head and neck from the outside in. We had to kind of convert that to an inside-out approach and learn the anatomy from an endoscopic standpoint. And so the field had to change, and then, as I'll explain to you, we had to figure out what quality surgery was to try to roll this out and perform clinical research like our colleagues have done. So I'll go kind of quickly through this. Many of you have probably heard about the transoral robotic surgery. The surgeon sits here in the console. The devices go through the mouth. The resolution and the optics are quite good. The instrumentation is wristed 540 degrees, even better than God made the human wrist, so we can really get into corners and get these tumors out. So this led to an FDA approval in 2009, quite a while ago, without any prospective data. There was simply safety data. And so then fortunately about a year and a half ago, we had a second technology FDA approved for head and neck cancer. We had the first device I think, in the country and have been using this. It's not really a robot. The surgeon sits at the head and manipulates this, but it has a flexible optic, so you can see around the corner, even down to the larynx. Here are the vocal cords. And you can see how the operating and cutting devices are here with the endoscope, can go in through the mouth and can operate. And so now we have a little competition. We've got two devices, but we really don't have any data. Where does this fit on our category of choices? Do you get an operation simply because you walk into a center that uses it? And what data do they rely on when counseling patients? So Drew Ridge, during his presidential address at the American Head and Neck Society, challenged the surgeons. He says, we, the surgeons, show anecdote and pictures. Our colleagues show curves, Kaplan-Meier curves and data. And so surgeons need to learn data. So that's what led many of us to try to get together and rise to this challenge. So what's the rationale for de-intensification surgical trials? Well, as I mentioned to you, surgery can potentially personalize therapy. I told you about tumor bulk. Well, if we don't think radiographic imaging is the best, perhaps upfront surgery can tell us about pathologic risk factors. The neck dissection can tell us the number of lymph nodes. And so we needed to reevaluate all of this for the HPV positive subset of disease. And we have FDA-approved devices, but we have no oncologic efficacy data. And we need to have this in order to offer it to our patients. In fact, I think it's somewhat immoral to use a device that the surgeon gets paid for using without data to support its use when you have a 95% cure rate with radiation or chemoradiation alone. But we needed to design clinical trials with a slightly different approach. It was not going to be an improvement of efficacy. It was going to have functional endpoints and so on. So we needed level 1 data. And then, and I'll talk a little bit later about the Cancer Genome Atlas. TCGA did all the genomic analyses of HPV positive and negative disease. But even though we've had a 3-fold increase of HPV positivity in the field, TCGA only had 10% of the tumors that were HPV positive. So we've got a totally new disease, but none of our tissue banks, none of our freezers had HPV positive tumors because we weren't operating on this disease. Now that's not a reason to run a surgical trial, but the trial I'll describe to you will create a repository of 500 HPV positive head and neck cancers available to the community at large to study this new disease, and I think it is one side benefit that we'll get from a surgical trial. So the Eastern Cooperative Oncology Group, with a clinical trials planning meeting, the NCI has a clinical trials planning meeting, and I brought a single-arm phase 2 trial to the clinical trials planning meeting, and it emerged with a randomized phase 2, so the meeting really helped. This is a trial of upfront surgical resection for resectable HPV positive disease. You know, P16 is the surrogate biomarker. It's the intermediate bulk of disease for which the robot is FDA approved. And we stratify by tumor bulk and smoking status. Now what we had to do was we had to get all the surgeons on the same page because the upfront surgery is FDA approved. That's not the investigational component. So we created a credentialing process where we've had about 125 applications. We've credentialed 78 surgeons. They have to submit pathology reports and their surgical cases. In fact, tonight we have another teleconference, and this is now open at 60 sites where there's at least one surgeon. Some sites have 2 surgeons credentialed. We had to get the pathologists on the same page. How they gross out a specimen is different from center to center, looking for extracapsular spread and so on. And then Jan Lewin and Kate Hutchison at MD Anderson harmonized and synchronized our speech and swallowing, our modified barium swallows, because the modified barium swallow is a way for us to see what is the impact on function in this trial. And they get one pre-surgery, and then 4 weeks later before they start radiotherapy, and then at 6 months, and then at 2 years. So we will have a lot of swallowing assessments similar to what the HN002 is doing. So we'll have a lot of good data for the field. So patients get upfront surgery. The ultimate in de-intensification, about 11% of the patients so far get nothing further. This is arm A. And 40 to 50 patients so far are under the observation arm. Similar to the stopping rules in the HN002, we have a stopping rule here where if more than a single recurrence happens in arm A goes away. So obviously arm A is still running, so the observation arm appears to be functioning. Then we have the randomization where the statistical power comes in, margins less than 3 millimeters, extranodal extension that is microscopic, up to 4 metastatic lymph nodes. These are intermediate risk features which lead to randomization of the patients between 50 gray and 60 gray. And then we have a high-risk arm, which is about 30%, up to 33% at different points in the trial, where we're not comfortable de-intensifying. Extensive extranodal extension in the lymph nodes, or 5 or more metastatic nodes. This is the frustrating arm. I'll give you a new trial at the end, but this is about a third of the patients that are not getting de-intensified. In fact, they're getting 3 modalities. But we have passed the DSMC, and we have another one actually this month. Our 2-year progression-free survival is pegged at 85%, and so this trial is ongoing, and in fact, hopefully we'll finish accruing in the early summer. It's putting on about 15 patients a month, and we've been quite active in Pittsburgh as well. So we're very anxious to have this trial along with the HN002, and discussions are ongoing for where the trial would go.
Why do you consider the lower arm frustrating if it's high risk?
Well, great question, because we actually don't know that they're high risk. It makes sense that they're high risk, but we don't have so much data for HPV positives.
For non-HPV positive.
Absolutely, completely agree. So for the new disease, what I would say is some data suggests 2 millimeters of extranodal extension is the true prognostic factor. There's a paper coming out in Cancer where the group took the National Cancer Database, and between the gross extracapsular spread, there was about a 4% difference in the HPV positives. It was like 93 versus 89%. Still pretty darn good. And as you know, the mock HN, 16,000 patients in the Pignon showed the addition of chemotherapy only adds 4 to 6%. So if the prognostic impact is only a decrement of 4 to 6%, you know, I think we have to weigh whether, so in the trial I'll describe later, we're not adding cis. I think we need to add something, but maybe not something toxic. And when you enroll somebody, as was brought up by this counseling, you enroll somebody on a de-intensification trial, everybody thinks they're going over here, but a third of them are going to end up with 3 modalities when we know they could have done pretty darn well with 70 gray and cis. Now they're getting 4 less gray, but an operation. So I'll tell you what we're doing with that. So surgical technology is moving. We're finally going to get some data. I hope that I'm not going to be the goat of the American Head and Neck Society providing level 1 data that you should not operate on oropharynx cancer. Hopefully it will not have hurt patients, but we'll have to see how it reads out. The question is, where does it fit? And so I would suggest to you as we transition to the 4th modality is that we've sort of maximized our ability to rearrange and reassort combinations of the traditional modalities of surgery, radiation, and chemo. And for years, the targeted therapies field was to understand biology and try to target changes at the protein level in the tumor cell. And those folks got the plenary sessions, they got the big auditorium. The immunotherapists were in the basement. We got the stale muffins, and the lights were flickering. We were curing a lot of mice. And then what happened was I think everybody began to understand that the tumor microenvironment was important, that even traditional chemotherapeutics may be affecting the infiltrating host immune response, and it's become really one field of targeted therapy and immunotherapy. And many radiation and chemotherapy may actually have an immune mechanism. So this is quite an interesting development, but I would suggest we now have a bona fide 4th modality, as I'll explain. But I first need to convince you the importance of the immune system. This is Jerome Galland's work in Paris. This is the European staging system, where stage 1 does well, stage 4 does poorly. That's not a surprise to anybody, but you can reassort this prognostic staging completely by the extent of infiltrating immune cells. And so no matter what the stage is, if you have a high density of tumor-infiltrating lymphocytes, patients do well. No matter what your stage is, if you have a low density of tumor-infiltrating lymphocytes, the patient does poorly. So there is not just biology in the tumor cell, but there's the ability of the host, the patient, to see something abnormal going on. And the goal of immunotherapy is to convert this low TIL into a high TIL scenario. Sir?
If
This is colorectal cancer. So thank you for bringing that up, and I should have qualified that. This is colorectal cancer, but it has been shown in head and neck cancer. The head and neck field is a little bit flawed, in my opinion, based on this. This is called the immunoscore, to quantify the density, in part because the HPV positives occur in a lymph node. The tonsil is a lymph node. And so it's always challenging to me how the publications for the immunoscore in HPV positive disease integrate the fact that the tumor is invading from the epithelial lining into the tonsil, which is a lymph node, and making sure the pathologist has accurately counted truly tumor-infiltrating lymphocytes as opposed to sort of bystander lymphocytes that the tumor cells infiltrated into the lymph node of the tonsil. And so I think it's a little flawed, but there are data in HPV positive that it affects prognosis. The problem we have is that HPV positives do so well that it's hard to do prognostic biomarkers. But this is colon. It's been shown in breast, lung, on and on. So this is a general phenomenon of host immune response. But a good question, thank you. So what got many of us started is that the targeted therapies field had the goal of sort of using a drug, and then when we ran the trials, vemurafenib for melanoma is a great example. We get great immediate responses, and then you understand the biology of tumor heterogeneity because eventually resistance occurs. The strength of the immune system is memory. All of us got our vaccinations as kids, and this tail, the durability, is what we're all hoping for. And so when we can get such a signal, which we have in many cancers, and I'll show you some data in head and neck, then can we move this into the upfront setting for those HPV negative patients, and can we be more targeted with the HPV positives? So you have to know a little bit more about how the immune system works. And I know this is review for some of you, but it perhaps a bit of a mixed audience. So the way the immune system works is the first thing, it's like driving a car. This is from Jed Walchuk at Memorial, who helped us kind of conceptualize. You got to turn on the engine. You need a baseline immune response, an antigen-specific response. And that can be an endogenous response. It can be induced by a vaccine or adoptive cell therapies. Then you need to boost it, step on the gas. You need to drive it with cytokines or inflammatory agonists, or we have some immunotherapies. And then what will probably get the Nobel Prize is the recognition that the immune system has multiple redundant checkpoints, negative regulatory signals, because what you don't want is an immune response to continue proliferating and going on once the antigen has gone away. And so these negative co-inhibitory signals called immune checkpoints are what the cancers take advantage of to turn off the anti-tumor immune response. So you have to take off the brakes to get the car moving, to get the immune response to proceed. So you need to know a little bit about the T cells. They're stimulated by dendritic cells to trigger that signal 1. And there are a lot of pathways within the T cell that we can trigger with immunotherapy. There are not only negative regulatory cell intrinsic mechanisms like PD-1 and CTLA-4, but there are negative regulatory cells like the Treg, these suppressive cells that in a cell extrinsic fashion can affect the strength of the immune response. And so a number of these can be targeted in the clinic as we understand them, and I'll show you some examples of overcoming immune suppression. So where we start in clinical research, of course, we start in the population where we can be most creative and most high risk, a group that has no chance of cure. The goal then is to move it into the locally advanced stage where we want to elevate the survival curve with durability and eventually see immunotherapy not only go to early stage disease, but immunoprevention, not just vaccines against HPV, but actually pre-malignancy. And I think we're getting in that direction. So now back to head and neck cancer. We have HPV negative, we have HPV positive. How does immunotherapy fit? And what I use this slide to sort of remind you is that it's all clear to our scientists that the tumor cell has vast differences in the tumor suppressors, in the oncogenes, either viral or genomic. But what we haven't really figured out is the immune escape mechanism the same between HPV positive and HPV negative? And so that's what my lab has been focused on for some period of time, and I'll give you some data from that. But I think we need to understand to use immunotherapy, are the immune escape mechanisms the same? So, gosh, I feel old when I show papers we published 12 years ago now, but we just asked a question. A postdoc in the lab, Andreas Albers, asked, is there a baseline immune response to HPV in cancer patients, head and neck cancer patients? Looking at HPV 16, the E7 protein, and quantifying the T cells in the peripheral circulation, you can see that there is a group of HPV positive T cells that is higher than the HPV negative or the healthy donors. So these cells exist, virus-specific T cells, but they're not able to control the cancer. These are tumor-bearing patients. So we had to kind of go back and figure out what is the escape mechanism. Well, signal 1, turning on the engine, as you can see here, is the T cell receptor seeing its ligand, the HLA peptide complex, in this case an HPV peptide. And so we had the hypothesis from collaborations with Soldano Ferroni when he was in Pittsburgh, and the downregulation of the HLA peptide complex could be a mechanism to evade T cell recognition. So we wanted to gain some data in that. Then we wanted to look at the PD-1, PD-L1 expression. This is the signal 2 that I'll describe a little bit more later. And we stained, this is double staining. You don't need to know which is brown, which is pink, but one's the receptor, one is the ligand. And what you can see here is that there's 5 different patterns. Some of these, there's very little PD-1 receptor and PD-L1 ligand. But then this one jumped out at us, where you see PD-1 and PD-L1 almost overlapping like a synapse, where you have the receptor and ligand. And this indicates a true immune escape mechanism. And in fact, the fact that 1 of 5 of these tumors has this very co-localized PD-1 and PD-L1 would suggest to me and mirrors the 20% response rate that we see in the clinic. So PD-1 may be present, but maybe it's not pathogenic as an escape mechanism. So back to the processing pathway, can we get any evidence for HLA processing as an escape mechanism? So it turns out that this operates in series, where antigens get degraded by the proteasome, they get pumped in by the transporter associated with antigen processing, and eventually they get out on the cell surface where the MHC peptide complex gets presented to T cells. So if a tumor cell can block any step in this pathway, then it can escape an antiviral or tumor-specific T cell. So we looked in a series of HPV positive head and neck cancer patients. On the left, you can see staining for the proteasome, for the TAP transporter, and for the HLA class 1. The surrounding mucosa stains very nicely. When you get to the tumor cells, it's downregulated all these steps in the antigen processing pathway. And when you say, well, no, I see some staining for HLA class 1, this is actually the fibroblasts in the stroma. The epithelial cells have all lost HLA class 1 at the cell surface. And if you transfect one of these, TAP, the peptide transporter, you can restore recognition in an HLA class 1 restricted fashion, suggesting that you can reverse immune escape by putting the antigen processing machinery back into this cell. This is an HPV positive cell, a head and neck cancer cell. And that was in vitro data, but when we got to the Cancer Genome Atlas, and my contribution to this effort was to focus on immune escape pathways that were mutated. What you can see in the box is that the HLA and interferon pathways are quite highly mutated in head and neck cancers. And in fact, on the right is the HPV positives. On the left is the HPV negatives. There are some differences, but it's a pretty general mechanism that you see alterations in the HLA beta-2 microglobulin. And although these may seem like relatively low-frequency events, Tony Rebus published in the New England Journal last summer that resistance to anti-PD-1 therapy is associated with acquired mutations in beta-2 microglobulin and the interferon pathway. So these are bona fide escape mechanisms, not just in vitro, that will be barriers to the immunotherapies that we're delivering to our patients. And in some of the trials that we've run in head and neck, we're analyzing this at the genomic and the protein level. So that's signal 1 escape. Now let's go on to signal 2 escape, that taking off the brakes, because the PD-1, PD-L1 interaction is the immune checkpoint that applies the brakes, trying to slow down that immune response that we want to take off the brakes and reactivate. So PD-1 positive cells have been said to be exhausted because PD-1 goes up when you're exposed to the antigen. And so the problem with HPV, unlike an acute viral infection where the antigen goes up and then the antigen goes away, with HPV, the antigen stays there, and the T cells in the microenvironment are seeing HPV antigens repetitively, so the PD-1 positive T cells get progressively exhausted. So from a series of tumors that we got from the operating room, this is how our lab is driven, is that tumors come over every day, get digested, we analyze the tumor-infiltrating lymphocytes, and we did a pretty simple thing. We just counted the number of PD-1 positive T cells in the microenvironment in the tumor-infiltrating lymphocytes versus the peripheral blood lymphocytes from the HPV positive on the left and the HPV negative on the right. And a pretty surprising finding was that the HPV positive patients had a higher frequency of PD-1 positive T cells in the microenvironment. This is the good prognosis group. So in a paper that is hopefully coming out somewhat soon, what we did was looked at the extent of PD-1 expression. So it's important not to just quantify a binary PD-1 positivity, and the highest PD-1 expression was in the HPV negative patients, and you can actually show a relative odds ratio for recurrence associated with the extent of PD-1. But what this shows you is that PD-1 is highly expressed in the microenvironment. It might imply that anti-PD-1 therapies could work better for HPV positive patients because there's more of the receptor there to block. And if you look at the ligand, as we published last year, the ligand is present at about 60% frequency in head and neck cancers, more often in HPV positive tumors. You can see the expression here. And what we've been trying to do is understand the regulation of PD-L1, in part because this will help us to understand resistance, but also I think it's a good example, and as we showed last year, the EGFR actually drives PD-L1 expression through the JAK-STAT pathway. So this would suggest a good link between an oncogenic signal, we think of EGFR driving tumor growth, proliferation, anti-apoptosis, but actually EGFR can drive immune escape through pushing the PD-L1 expression and immune escape. So EGFR inhibitors, therefore, would be potentially good partners with anti-PD-1 therapies to overcome immune escape. So I'll go somewhat quickly through data that you probably already know. This was a trial that Mara Gillison and I ran with Bristol Myers. This was a phase 3 trial applying an anti-PD-1 therapy to ask, is there a signal in a recurrent metastatic population? And in data that we've submitted to ASCO, we have now prior cetuximab exposure, an EGFR inhibitor, to understand whether patients treated with nivolumab randomized versus the single-agent investigator's choice chemotherapy could do better or worse. But this was published last fall, and in fact, while we were at the SITC meeting, nivolumab was approved for head and neck cancer. Pembrolizumab, another anti-PD-1 therapy, was approved in August, so we have 2 antibodies now approved for head and neck cancer. And we had an overall survival endpoint, so we don't have to rely just on response rate. We can actually look at the number of patients alive at a year, and there is a more than doubling with anti-PD-1 therapy versus the standard of care systemic agent in the comparator arm. And the kind of Goldilocks of this is that the adverse events, the grade 3 and 4 adverse events, were 1/3 the rate in the setting of a doubling of a 1-year survival. And Kevin Harrington has just recently submitted our quality of life data that the quality of life in the nivolumab-treated patients, in fact, stabilized or improved, whereas the standard of care arm declined consistently. So you have a nice agent that can improve survival with a much lower rate of adverse events. And the question is, where do we go with this? I just want to show a picture. Everybody likes to see tumors going away, but it reminds us why quality of life is an important endpoint in these. And I think what we don't have in the immunotherapy field is good value metrics. You can see it on an anecdotal basis, and I'm sure for this patient it's worth $150,000, which is the cost of nivolumab for a course. But I think we do need to have better metrics to quantify this for society. So what about biomarkers? What about HPV status? Well, you can see that the curves begin to separate very early, at 1 to 2 months in the nivolumab-treated patients. And the hazard ratio of 0.7 for the overall trial improved to 0.56 for the HPV-positive patients HPV negative patients still did well, hazard ratio about the same as the overall trial, a 27% reduction in risk of death. But the HPV positives in this trial seem to do better, and you can begin to see this tail of durability, which will, as I mentioned, be further presented at ASCO with the one-year follow-up data that Mara will be presenting. What about PD-L1 as a biomarker? Again, the curve separating early in the PD-L1 positives, hazard ratio of 0.55. What do we do with the PD-L1 negatives? This is really one of the issues in the field because you can see that these curves appear to separate, but late, 8-9 months. There appears to be a bit of a tail. The hazard ratio modestly favors nivolumab, but quite liberally crosses 1. So what do we do with these patients? I will tell you that this holds up. In fact, it improves a little bit in the one-year follow-up. But is this sufficient in a PD-L1 negative patient? What I will tell you is that of the 6 complete responses in this trial, 2 of them were PD-L1 negative, meaning less than 1%. So 2 of those patients got tremendous benefit, and excluding based on PD-L1 status at present is not very satisfying. So we need therapies to make this group more responsive. So what are those therapies and where's the field going? Well, I think many of you know from the melanoma and our non-small cell lung cancer group, targeting with CTLA-4 is promising in combination with PD-1. There's a number of agents to do that. And then there are all of these positive signaling molecules. And so what are the combination therapies at present in head and neck cancer? There are these 2 phase 2 trials combining PD-L1 with CTLA-4. There's 2 phase 3 trials, one in first line called Kestrel, then one in the second line similar to the Checkmate trial, combining dual targeting versus PD-L1 monotherapy versus standard of care to try to ask, can we get better response rates? Well, what's the data to expect that? There's melanoma data that Jed Wolchok showed, and there's lung cancer data that was at last year's ASCO. So in a subset of patients, you can actually see that combining anti-PD-1 with anti-CTLA-4 does have improvements in response rates, although not in every patient. So I think the head and neck data, which will be emerging, those big 700-person trials I just showed you in head and neck, have put on about 500 patients. So I think pretty soon we'll have combo data for head and neck cancer as well. So we have all of these positive co-stimulatory and then negative co-inhibitory. How do we figure out what combinations are best going forward? Well, we had an interesting finding. Many of you know in immuno-oncology, the companies will open up a phase 1 multi-tumor cohort. And it turns out that targeting a unique immune checkpoint, the killer inhibitory receptor, which is on the natural killer cells, could be targeted with lirilumab. And we've been talking about T cells, a lot about T cells, and all these cell-intrinsic signal 2s, like PD-1, CTLA-4. But don't forget there are these extrinsic cells in the microenvironment. The immune infiltrate is quite complex. So the hypothesis here was natural killer cells can drive a strong so-called type 1 immune response. We're kind of partial to NK cells in Pittsburgh because our founding director, Ron Herberman, discovered them at the NCI. And so we love NK cells. So combining nivolumab with an anti-KIR to activate natural killer cells was quite appealing. So we participated in this trial. Ram Leidner out in Portland presented these data at the SITC meeting last fall. What you can see here is that head and neck was one of the tumors in the phase 1 cohort. And so it turns out that the only promising tumor type was head and neck cancer. We've thought that it's an NK dependent tumor for quite a while, so it was kind of fortuitous. And we took these patients out and reported them. And what you can see here is that a pretty impressive spider plot with deep and durable responses. This is a small cohort, only about 25 to 30 patients at this point, with further data coming down the road with longer follow-up and more accruals. But you could see a number of these patients doing quite well, some of them bouncing back and forth, but looking like they're going to respond. And we did something, I hope there are no statisticians in the room, but we sort of put it up against the Checkmate trial, which was monotherapy. I will tell you that the eligibility were identical. They had to progress within 6 months of platinum therapy, and the demographics indicate a similar performance status and so on between the populations. but the Checkmate trial had a 13% response rate, and combining nivolumab with the lirilumab anti-KIR had about a 10% improvement in response rate. Interestingly, it was unique only in the PD-L1 positives. So what we find with these agents is that we learn the biology while we're trying to help patients. And so you can see that there's increasing responses the more PD-L1 that's present. Okay, what are the other targets? What's coming down the horizon? Well, we have a new grant looking at TIM-3. You can see staining for PD-1 on the tumor-infiltrating lymphocytes versus TIM-3, that there's a lot of double positive cells, up to 50% co-express PD-1 and TIM-3. And in fact, I told you the highest PD-1 is the worst, most exhausted cell. You can see that the TIM-3 positive, double positive cells have the highest PD-1 expression. So these are the cells we want to target. And so we can use flow cytometry to show in red here that these are the most exhausted cells. These are genes associated with exhaustion. You can see that these cells make the least interferon gamma, so they're real duds when they express both PD-1 and TIM-3. And when we actually treat in vitro with an anti-PD-1 antibody, you can see that the TIM-3 goes up 2 or 3-fold. So it's an escape mechanism if you use monotherapy. So then we went into a mouse model of HPV positive head and neck cancer. We treated with anti-PD-1, and then you can see the tumor-infiltrating lymphocytes in the anti-PD-1 therapy really expand the TIM-3 positivity, suggesting, and which we've shown, and it's actually now published in Oncoimmunology, an addition of an anti-TIM-3 to an anti-PD-1 therapy does have improved therapeutic benefit. So understanding the escape mechanisms of monotherapy is where we need to go with specimens. So just in the time I have left, I want to tell you about where we are with the advanced stage curable disease. I want to remind you that EGFR is still an important prognostic biomarker. Dong Xin and others in Georgia here have worked on EGFR and its role in driving tumor growth and its the role of therapeutic targeting. But what we have is an EGFR that's present in over 90% of the cancers, but response rates in only 10 to 20%. So how do we figure this out? So the lab has been focused on the immune mechanisms of EGFR inhibition. I showed you that PD-L1 could be overcome by blocking EGFR, but antibodies, cetuximab, targeting EGFR can actually trigger an immune response through antibody-dependent cytotoxicity and stimulation of T cells. So we needed to prove that to everybody. So in our previous SPORE, Project 3, we ran a prospective trial of cetuximab, and we got specimens pre and post to try to understand the immune biomarkers and enhance cetuximab's activity. So you can see what a responder looks like. About a third of these patients responded to a month of cetuximab, and then the other 2/3 did not. I always feel bad because I think this tumor grew just a little bit over that month, but we were able to resect it still, collect specimens, and then figure out responder, non-responder status. So it turns out the non-responders upregulated the regulatory T cells, these Treg, as you can see in the peripheral blood and in the TIL. They, in the responders, did not upregulate these CTLA-4 positive Treg. So this is a targetable marker on Treg, so we can combine cetuximab with an anti-CTLA-4 antibody. Our current SPORE is focused on a clinical trial that we finished up last summer. Julie Bauman presented at ESMO the safety data, and I've submitted for ASCO on the oncologic efficacy data, which looks quite promising, to combine cetuximab radiotherapy as one of those FDA-approved regimens and intensifying it for high-risk HPV negative and HPV positive patients, smokers, heavy tumor burden, to try to, but you see here we've eliminated chemotherapy. We've got 2 different antibodies, so we're targeting the tumor or we're targeting the immune system to try to enhance the activity. And we think radiotherapy, fortunately, Wally will be happy about this, is a good immune partner. What the right dose and what the right sequencing is things we're working on in the laboratory, but I think driving tumor cell death and antigen transfer is a good thing. With Maura Gillison and Quinn Lee in the RTOG Foundation, many of you know this trial is now launched. We had a call last night about the phase 1 portion, which is just about done, and we should be launching the randomized component of this trial. It's a busy slide, but this will be combining nivolumab, anti-PD-1, with chemoradiation in different schedules, with cetuximab, or in patients who are chemotherapy intolerant. And so I think this will hopefully be a practice-changing trial as we get other data to suggest that we can intensify therapy. What we've asked in Pittsburgh with Julie Bauman, Jonathan Beitler is the radiotherapy co-chair on this trial, is should the anti-PD-1 be during or could it be after chemoradiation? So patients get randomized to either concurrent chemoradiation with an adjuvant pembrolizumab, or they get a loading dose of pembrolizumab, which in our laboratory we've shown is actually key, getting the anti-PD-1 on board prior to the radiation, and then give pembrolizumab as a concurrent therapy to sort of prevent some of the immunosuppressive effects of chemoradiation. So this trial is ongoing, accruing pretty rapidly, and we're very interested in opening up here with Nabil at Emory, and Julie has moved on to Arizona, so we'll have a few sites. So where do we go from there? Well, let's not forget that we have a vaccine available. In fact, Genter published for vulvar intraepithelial neoplasias, and they have an update in cervical cancer in Science Translational Medicine that you can vaccinate against HPV therapeutically. So we have a new investigator-initiated trial with Merck. They will get this ISA peptide vaccine, and then they will get pembrolizumab. Then they go on to chemoradiation, where we give additional vaccines plus pembrolizumab. So we can boost that HPV-specific immune response and prime it while we're using anti-PD-1 in combination with chemoradiation. And in this paper last year, we showed that it's important to get the vaccine prior to the anti-PD-1 because otherwise you sort of dampen the vaccine effect. And then the last trial I'll show you, coming back to that high-risk arm in the Econ trial that, as I mentioned, I wasn't too happy about, is to ask a question, can we de-intensify or be more targeted toward the high-risk HPV-positive patients? So all patients will be in that high-risk group. The heavy smokers, the heavy tumor burden, N2C, N3 necks, 5 lymph nodes, and they will undergo surgery of any type, all HPV positives. And then what they'll get is a 50 gray in 4 weeks, the so-called DeHanka regimen. And Adam Rabin has been very helpful in designing this trial with me. BMS will fund it to do upfront surgery, nivolumab as opposed to cisplatin, and a 50 gray accelerated fractionation in the hopes that we can be more targeted with the population and de-intensify some of the radiotherapy, but do it in a way that integrates a more effective therapy from a functional standpoint for a high-risk group that probably doesn't need all 3 modalities. So the key is to target the tumor microenvironment. The role of a surgeon, I think, can help to deliver specimens to colleagues, either in our own labs or in collaboration, and try to understand why some of these TIL don't get into the microenvironment. We have to understand all of the blocking inhibitory mechanisms in order to reprogram them. And then we can't forget the barrier, as I mentioned to you from TCGA, the barrier is the tumor cell has been selected to be a poor antigen-presenting cell. And this is likely to be a problem we're going to have to overcome with a subset of non-responsive patients, and how to avoid the escape mechanisms that Tony Rebus showed us, which are mutations in the JAK-STAT pathway or beta-2 microglobulin, which are acquired resistance. And we always see the melanoma folks publish it first, and then we have to look for it in head and neck and other tumor types. So HPV-positive cancer is rising dramatically. I think personalizing can be with surgery, with appropriate radiotherapy doses, and integrating immunology for the targeting of this new disease. We have to understand that immune escape as we're doing this with clinical specimens, and I think immunotherapy will become one of our standard modalities and already is taking its place. So here's the lab. My collaborators, I've been fortunate to have some support for this, and have enjoyed collaborating with the folks here at Emory. Be happy to take questions. Thank you.